Good day, grade 12 learners. Welcome to today's Life Sciences lesson. My name is Sina Sekheche, and this lesson is brought to you by the Free State Department of Education in collaboration with Pagama Research and Development. Right, so we are still continuing with our revision, but today's lesson is going to be a different one. Remember that in life sciences, you still need to study the skills. There are so many skills that are interwoven in the question paper, and most learners are losing marks when it comes to the skills. So today we are going to refine the skills in life sciences. That includes the skills of the scientific investigations, the graphs, diagrams, and tables. There are rules to answering questions on this, and we see that you lose marks on these. So let us go straight into it. For the practical investigations, you know that we have variables. This word variables comes so many times in the question paper that you will be asked to identify the variables, whether it is an independent variable or dependent variable or a control or a fixed variable. Take note that control or fixed is used interchangeably. Right. So when planning the investigation, you need to set these variables or the possibilities, and then you test them one at a time so that you can be sure that this is the factor or it is a variable that gives us the results. For an example, you cannot test heat, temperature, or light at the same time for whatever result that you want. So if you are setting an experiment, there has to be that one factor that you are investigating. Then you will decide whether you're going to limit or to introduce that factor to check its effect on your investigation. So the investigation will always have a factor, one factor that is going to be investigated at a time. However, there are many factors that can be part of that investigation, whether independent and dependent. Right. Each factor is known as a variable. So these factors that you are investigating are known as a variable. And each investigation should test only one. So we've already established that one. Generally, a scientific investigation will have an experiment and a control. So an experiment will be the one where you do not have the factor that you are investigating. And then a control will be the one that is going to give you a positive result. So with that factor that you are investigating. If, let's say, you want to investigate the effect of light on the growth of plants, then in the control, you are going to have light and all the factors there to see that growth. And then in your experiment, you will limit the light or not have light in the experiment so that you can investigate that factor as light. So that is how a practical investigation is structured. You must have your factors and have one that you are investigating, and that one is called a variable. It's a variable. Now, some of the terminology that you need to look at when you are studying these variables, there are three types of variables. The independent variable. Remember, it's one of those factors, but these ones are independent. So what do we mean by an independent variable? In an investigation or in a question paper, you may be asked to identify independent variables from an investigation. So what are these variables? The independent variables are the variables that are controlled or changed by the investigator, meaning you as the investigator, you have control over these variables. It's either you can limit or you can make it more. So it is up to you to change it. So you have full control on top of or on these variables. They are called variables. So you are going to determine what effect it has. So if you want to determine what effect does light have on the growth of a plant, you will decide, you will control the amount of light, or whether you introduce the light or you don't introduce the light. Then it is you who is controlling that variable, and you are going to check its effect on the plant. And then when you are drawing a graph, the independent variable that is shown on the graph, it is shown on the horizontal axis or on the x-axis. So when you have your graph like this, on this axis here, this is where you are going to find your independent variable. I hope now you understand what an independent variable is. And then you come to the dependent variable. Another question is identify the dependent variable. What is the dependent variable? It is the variable that is the effect of the independent variable. Like the results, the effect, what is happening. So that is the dependent, and it is depending on the independent variable. Then for our example that we mentioned above of a light with plant, then you can check that, okay, with more light the plant will grow more, and then you can record the growth in length of a plant weekly or daily or whatever method that you want to choose depending on your environment, on your experiment. Then you can record that information and say, during the course of 6 months, our plant grew in this much length in the intervals of 1 week, then you are able to see. So the growth was dependent on the light, whether you exposed it to light and how much did it grow. So you see, the dependent variable is that one where you see the effect. Now, this is responsible, or this is a response that is measured or monitored during the investigation. So you are going to measure the response of a plant. How does it respond to light? So that response that you are getting, it is your dependent variable. And then in a graph, your dependent variable is going to always be on your y-axis, on a vertical axis or on the y-axis. So now you need to be able to identify them and be able to draw them on the graph. Then we come to the last variable, which is the fixed or the constant variable. Now, these variables are there in any experiment, because when you set your experiment, you want your results to be credible. You want your results to show that you followed the correct procedure. Now, these fixed variables are the ones that they are all the factors that must be controlled or fixed or kept the same when conducting an investigation. An example can be the time that you allow the plants to be exposed to the sun must be the same, or the type of plant that you are using must be the same. So you see, it sticks to the same to keep everything constant, the ones that are all going to be the same in an experiment or in different experiments. So you don't want them to use this type of plants to check for light and use a different type of plant, because then your investigation will not have conclusive results, because you are not using the same. You did not keep your fixed or constant variables in check. So this one, when you want to remember the fixed or constant variables, remember they are those ones that you have to keep the same. Right, so that is the terminology that you need to get on. Then we come to hypothesis testing. What is a hypothesis? I know that you know what is a hypothesis. However, the way you formulate a hypothesis, that is where you might be getting it wrong. Right, a hypothesis is an attempt to explain some event or observation using whatever information is currently available. So you are trying to explain what you are seeing or what you are observing, using whatever information that is available to you. So you give us an explanation without having studied it. So it can also be regarded as an educated guess, like informally. So it's an educated guess. You guess why a phenomenon is in this way or why a situation is in this way. But when you are doing this guessing or predicting, you don't write it as I guess the reason why this is happening is because of what. You write it as a statement that you are sure of. That is what makes a hypothesis to be real. And a hypothesis must be testable. You must be able to test it. Whatever that you are saying, the other person must be able to conduct an experiment to test your hypothesis and later be able to reject it or to accept, to say it has been accepted or it has been rejected with proof of the investigation of or the results or the interpretations of the results thereof. So in your hypothesis, there must be two variables when you formulate your hypothesis. So you must tell us that the growth of plants will be more or will increase when it is shown to light or when it is exposed to light. The growth of plants will increase when it is exposed to light. So you have growth of plants, which is your dependent variable. Then you have light as your dependent variable. Then you will do an experiment to test that statement that you are saying. So you see, I did not say, I guess this plant is growing because of light, even if I'm thinking it. But when I write it, I have to write it as a statement that I am sure of. So you must write it as a statement that you are sure of, as a full statement. Then you must state the relationship between the two variables, meaning when there is the other one, then it will influence the other one to increase or to become less. That is you now explaining that relationship. And it must be testable. If you are saying a hypothesis is like this, somebody must be able to set up an experiment to be able to test your hypothesis and come up with a conclusion and reject or accept your hypothesis. And then the independent variable must be the cause. The independent variable will be the cause. Hence, I said light will cause the plant growth to be more. So it must be the cause. And then the dependent variable is the effect. What is going to happen after once you introduce the independent variable? So one thing that I want you to remember is that a hypothesis is when you are trying to explain some event or observation using whatever information you may have available. And your hypothesis, you write it as a statement that you are sure of. However, it can be regarded as an educated guess. But this guess, you don't say I guess, you don't say I think, or I predict. You write it as a statement that you are sure of. Right. Then you come to another thing that you must know when you are dealing with practical investigation. This is reliability. You are always asked questions on reliability. Reliability, the idea behind reliability is that the researchers must be able to perform exactly the same experiment under the same conditions and generate the same results. So you must say, how do I know that this is reliable, or my experiment is reliable, or my results are reliable? It is when you know that other scientists can perform the same experiment under the same conditions and generate the same results. So if they are able to do that experiment of yours and get the same results, then you know that your experiment is reliable or your results are reliable. Now, in questions which ask learners to state how the reliability of the investigation could have been improved, the correct answers could generally be the following. Repeat the investigation. Now, before you used to just stop here, but in your answering, please check how many times it was done and write how many times they must repeat it. Because when you just say repeat it and you don't get it, you must write the full statement to say repeat the investigation at least 3 times and get the average. So you see, now you are telling us that that will then be reliable. Also, increase the sample size. The sample size speaks to number. If in your examination you were given that the scientists use 20 plants or 20 pigs, when you are writing that they must increase the sample size, state that number, that if you want to move it from 20 to 50, then you are going to get a mark to say they could have used 50 plants instead of 20. Then you are making it more practical to the practical investigation that is in front of you. So you don't answer in general. Then another thing that can help you to answer the reliability question is to say, take many readings and use the average of results. So this one goes with the repeating of the investigation. So when you repeat, you will say that the scientist must repeat the investigation and take many readings and then combine those readings to get the average. You use the average of the results. Then we know that these results, we can trust them. They are reliable because the scientist gave us many readings. They did not just give us one. So you take many, not one. Right. Number 4, increase the period of investigation. Also, here you must specify. If you were given that these plants were put in these conditions for a week, when you answer this question, for these results to be reliable, you can say the investigator could have put the plants for 1 month. So you see, you are increasing the period. So, but by being specific to what is in front of you, please make sure that you check what the investigation is saying, and what you are saying must be more in terms of the numbers that you see on your investigation. I hope you'll be able to answer now questions on reliability. Then the last one is select sample randomly. Because if you choose your sample based on, I'm just taking the plants in the front row, or I'm just taking the plants from this particular area, there might be some other factors that are advantaging or disadvantaging the plants. So if you take from different places randomly, then you are making your test to be even more reliable. So random sampling will also help to increase the reliability. So this is how you answer the question of reliability. Then another one is validity. I know that you confuse validity with reliability. Remember, reliability has to be, how do we trust? So validity, how is it valid? What did you do? Did you do the correct thing? So the validity questions, it questions the how. How? You see, how did you do your experiment? Or how did you carry it out? You check your conditions, your factors. Have all the factors or the variables being controlled or fixed or kept the same except the variable that is being tested? So when you know that this experiment is valid, the answers of validity must go around fixed or variables that are kept constant or controlled. Then you know that it is legit because the variables were kept constant, except for that one that you are testing. So is the design of the investigation appropriate? So once you set up your investigation, did you consider how it needs to be done? Is it the correct design, or did you do it in a lab, whereas you need to do it outside, where it needs the sun and other environmental factors, or you are doing it where it will be limited in terms of other variables? So the design, is it appropriate for the type of investigation that you are doing? This is answering validity. Also, when being asked about validity, you must answer around the variables being kept the same. You remember that one of constant variables. So validity deals with constant variables. You will talk about those variables that must be kept constant in the experiment throughout. Right. So hypothesis testing. Scientific investigation can also be regarded as a hypothesis testing, because when you start a scientific method, you will start with your hypothesis and then you design an investigation to test that hypothesis. Hence, it is also known as hypothesis testing. So when you start by observing phenomena. So this is the first thing. You will observe any phenomena in nature to say people who are tall, they have long femurs, or they have long humerus, whatever. You will just look at any observation that you want to see, or plants that are growing in a particular area, they grow more, or more people are able to roll their tongue, or whatever that you want to observe. It's just an observation in nature. It starts there. Then you come to number 2. From observing, you are going to ask your question about this phenomena. What is the question? You are going to ask yourself, why? Why is that happening? Why do people have blue eyes, or people who come from Antarctica have blue eyes? Then you will start your investigation. Or why are the learners, or 50% of the learners in my class, able to roll their tongues, whereas the others are not able to roll their tongues? So you see, it is a question now. After having this question, you are going to try to come up with what? You are going to formulate all possible variants. Now, through this phenomena, possible various various factors. From those factors, you come with a hypothesis. You come to say it is because they inherited a gene or what, or whatever. And then you formulate your hypothesis to say plants that grow in Antarctica, they do not have 1, 2, 3. They do not have roots. Let me just say that. Or they do not have what. You just come with your hypothesis and then you test that hypothesis. This hypothesis must be testable. It must be something that you are able to design an experiment or an investigation to test it. Right. So from this hypothesis, you are going to identify your cause and effect. Tell us that this is happening, which is the effect, because of this. Then you will tell us what is the cause. So your hypothesis will always have 2 variables. So this is talking, the cause and effect is talking about the 2 variables. Remember, we said we must see the relationship of those 2 variables in the hypothesis. Now, when you formulate this hypothesis or this prediction, you must follow this criteria. You must explain all aspects of the observation. So everything that is coming up with what you are observing, you must explain it in your hypothesis. A brief explanation to say, okay, this is what I am seeing. Then you can summarize that into 1 sentence. Then be the simplest possible explanation. Don't look for the deeper explanations that you cannot even test yourself. So if people have blue eyes, then you need to know that it is something that is genetic. It will require you to do a genetic testing, what, blah, blah. Then you have to give reasons. But for you now, you can have something like plants using those lights, or checking the amount of auxins, or on the effect of those auxins on the growth of the plants, on the growth of the roots, where do they grow. So you see, you must use the simplest explanation to say the plants will grow more on the side where the auxins are, or it will grow less on the side where the auxins are, whatever. Then be expressed in such a way that predictions can be made from it. So when you write your hypothesis, someone must also be able to predict from that hypothesis, and it must be testable. We cannot emphasize that more enough. So that is your scientific investigation, the backbone of a scientific investigation, where it starts. Then you start planning. When you plan your investigation, you must identify those variables to be tested. Then you must identify your dependent variable. Identify the variables to be kept constant. Same type of plants, same amount of time, same amount of water, same, you see, same everything, same. Check which ones you must keep the same, and then identify your independent variable and identify the list of appropriate apparatus that you are going to use to test what you are testing. So this is the planning stage of an investigation. So from this, then you can design an experiment and a control. Remember, we said the scientific investigation has an experiment and a control. So in the experiment is where now you know that you are going to limit your independent variable. Your independent variable, you are going to limit it in your experiment. And then in your control, you are going to have that independent variable that you are testing, so that the control can give you the positive results. Then you will check, how will you collect your data? select the method or the instrument that is most appropriate for the collection of this investigation. So these are the steps that you need to remember, because some of these questions, they will ask you to plan an investigation for a particular hypothesis. They will give you a hypothesis and then they say to you, write the planning steps of this investigation. So these are the things that you need to look out for. Decide on how you are going to collect or record the data. Are you going to use tables? Are you going to use diagrams? Are you going to use graphs? So that is the recording of the results. Then if you work with humans or animals, you need to get permission and ethical clearance. So you can't just take people and do an experiment or experiment on them without getting their consent. So it's very important for humans for you to get consent, or with the animals, get their clearance so that they cleared you to work with the animals. So these steps are what you are going to mention, because they are things that you need to consider when you are planning your investigation. Right. Then the results. Each and every investigation, after you've conducted it, you've done your experiment, there will always have to be results. You must give us the results. So you must decide. Remember, it said you must decide on how you are going to record the results. So this is how. Recording of the results or the findings, you must record the results in the form of tables, drawings, descriptions, pictures, etc. So it can be even graphs and is from the tables. Identify the patterns in the results. So here, identifying the patterns, you are analyzing. You are now analyzing the results that are in front of you. Identify and record irregular observations. So even those observations that you think maybe they might have impacted on the results, you must record them, because another scientist might use them or find them useful when they want to repeat this investigation. Also, reorganize your data. Plotting them in graphs is now trying to make it even more friendly and readable, so that another person can be able to just read your results and be able to interpret them. And also construct the flow charts or mind maps or concept maps to be able to read. So in this table, you see, you have light and dark. Then you see in one you have the values there. So if you put it like this in a table, it might not even be reader friendly. But once you put it in a graph, you can see that a graph is more readable. You can check that plant light, then you can read what is happening based on the data that you shall have been given on this graph. So then the last thing that you do in your investigation is to draw a conclusion. Remember, you have observed phenomenon, you have come up with an explanation, you've identified your variables, you have planned your steps, your planning and designing the experiment that is appropriate, you have recorded your results. Now you must come to a conclusion. So you draw a conclusion. Now, a conclusion explains the relationship between the variables, how and when the independent variables changes over time, the concentration, the pH, light, carbon dioxide extractor during the experiment. So tell us when what was happening, what is happening, by doing what now? By interpreting the results and remembering your planning steps. So from your planning steps, you can tell us, when I did this, this and this and this happened. So I can conclude that this and this needs this. You see? So you will be concentrating on the levels, the concentrations, the time, whatever that you are investigating there. Then identify and explaining trends or patterns from the graphs. So you will do that in your conclusion. Then also you can also do calculations in your conclusion. Explain the data. This is the analyzing that we were talking about. So in your conclusion, that data needs to make sense and you put a full stop to it. Then suggest specific changes that could improve the apparatus and technique. Sometimes something went wrong and it hampered your results and you were able to identify it. Identify it and suggest what can be done when this experiment is repeated. So that will be in your conclusion. And then also comparing data, the similarities and the differences. So let us take a look at an example of a scientific investigation. So I've been giving you different investigations in the explanations, but now we are looking specifically at one, right? So the first one, remember we said it starts with observation of phenomena. So here, a researcher observed that the potted plants he covered with black plastic bag did not grow. So you see, he covered the plants with black plastic bag. And then after that, he observed to say that those ones that he covered with black plastic bag did not grow. What is the next step? He must ask himself, why? Why? Remember, you come up with a question. And his why is going to lead to the answer, because his why needs an answer. Why are those plants that I covered with black plastic did not grow? Why did they not grow? Then he comes up with a hypothesis. After asking himself why, he will say, sunlight is essential for plant growth. Do you see that there is no way where it says, yeah, I guess, or I think? Because he covered them in plastic, they did not receive sunlight. But he does not say, I think sunlight is essential for growth, or I think plants need sunlight. He says it as a statement. So he says it as if it's something that he's sure of. Says sunlight is essential for plant growth. So this is the hypothesis. Then he must be able to test it. He must be able to now test it. Right. Going forward. Want to go forward. Then he must identify his variables. In planning the investigation, he set up 2 sets of 50 plants as follows. So you see this number is already telling you, remember where you have to increase the sample size. What are you talking about? Reliability. So when I ask you about reliability, you can say he can use 100. So when you say instead of 50 could have used 100, you see you are making it specific to the investigation that is in front of you. This is what I was talking about. Then you will get your mark. Then in the experiment, he put the 50 plants placed in a greenhouse. And then in the control, remember, he started by placing them in dark. So his is quite the opposite, because his control will therefore be the one that is placed in identical greenhouse but blackened, because this is actually what he was observing. This is what he was observing. So this must give him exactly what he was observing. But then he wants to investigate these ones that are not covered. What is happening there? Then, so that no light could enter. So this is how he set up his experiment. Then the experiment and the control differed in only one factor. That is, the variable is that plants were given light and the other plants were not exposed to light. I hope you were able to see that. So these ones, no light. These ones, there is light in the greenhouse. Right. So after this, what is happening? He knows. Going back. Okay. Right. Yes. We want to go back to the slide. So after setting his experiment like this, he is going now to let it sit for some time. He will decide whether for a week, for 2 weeks, for what, and observe what is happening with the plants that are exposed to light, what is happening to the plants that are not exposed to light. And therefore, he can go back to his hypothesis and check that, yes, I was correct. These plants, sunlight is essential for their growth, because the ones that were exposed to light, they were able to grow. However, the ones that were blackened, they were not able to grow. So you see, he was able to just do this simple experiment in life sciences as a scientific investigation. So that is what you can do. Now, the plants in both experiment and the control received the same amount of soil. Now, when they start talking about the same amount of soil, they are talking about the controlled variables, they are talking about the fixed variables, and these ones are the ones that make your experiment to be valid. So your validity is here. Same amount of soil, same amount of water, same temperature in the same room. So all of these are other variables that were kept constant. Now, in other words, they were given the same requirements, except for that one which was tested, which was light. Then he measured the growth of the plants over a period of time in the experiment and in the control. Remember, we did say. So then he found that those plants that were placed in the sunlight, they grew. Those that were in the darkness, they did not grow. So you see, from that, he can already draw a conclusion. Then he concluded that his hypothesis was correct for the type of plants that he investigated. So his hypothesis was that sunlight is essential for plant growth. And then he saw that those that were in the sun, they did grow. So it proved his hypothesis to be correct. So please make sure that you follow this, the investigation when you are writing an investigation. Then we are moving on to the next skill, which is how to draw a line graph. Sometimes you may be given data in an experiment in the question paper and interpret and analyze. And at the end, you will be asked to draw a graph. So there are different types of graphs that we are going to look at. The first type of graph that we are looking at right now is the line graph. So how do you draw a line graph? Right? First, you must identify the dependent and the independent variables from the information you are given. Usually, this is in a form of a table format. You will see that information in a table. In a table, identify your independent and your dependent, because in a graph, you will know where do you put your X, where do you put your Y exists. Now, the independent variable in the investigation, or is the one that the investigator can change. We've discussed this. The dependent variable changes as the independent variable changes. So this one, the dependent variable, is dependent on the independent variable. It will change when the independent variable changes. And the independent variable is the one that the investigator is able to manipulate. Then example can be time in hours in this example. Okay, the example that we are going to look at. Then the dependent variable, or it is a factor that is being measured. That is the temperature in degrees Celsius in this example. So we will look at the example further. So take a look at this. We were saying time in hours is our independent variable. Then temperature is our dependent variable. So the independent variable is usually given in the first column. Do you see this word usually? It does not say always. So the independent variable in this case, in terms of time in hours and the temperature, time is going to be our independent variable because it is on the first column of a table. Then the second column will therefore be our dependent variable. Then you know when you draw a graph, your independent variable comes on the x-axis, and on your y-axis, you have your dependent variables. Right. Then step number 2, draw a set of axes, or the x and the y-axis. The dependent variable goes on the y. This is what we were talking about. You see, time in hours is our independent variables. We saw that from the table. Temperature is our dependent variable, and they are on the y-axis, and this one is on the x-axis. So you have identified that. The independent variable is usually plotted on the x-axis. So we've identified that. Then, step number 3, choose a scale for the x and the y-axis. Make sure that your scale includes the highest numbers on the table of each of the variables. You do not use the values as they are. Example, 0 from 0 to 5. From 5, it's 9. How many spaces are here? So from 0 to 5, it's 4. From 5 to 9, you can say it's 4. 9 to 13. So you see, you do not use this. Here is 3. It does not balance. You must use the numbers in multiples. So, multiples. You must choose the scale in multiples. If you use a scale of 3, check your lowest number and your highest number. Then choose a scale that is going to move swiftly and appropriately between the two. So if you can move with the scale of 3, it can be 3, 6, 9, 12, 15, 18, 20. It will accommodate 0 and all these numbers up to 24. Then that can be an appropriate scale. So you use a scale of 3, 6, 9, 12, 15. You see, now you have same intervals here. You have the same spaces, or you are moving with the same interval. So when you choose a scale, include your highest number on the table and make sure that you have your lowest number, but you must use that in multiples. Remember this, multiples. Right. Then step number 4. Place the dots at the point where the two values for each result intersect or they meet. So you come and say, when the time in hours was 5, it is 5 here. Then the temperature was 24. If you go back to your table, you will see that it is 5 and 24. Then you plot a dot where these two lines are going to meet, or they are going to intersect. Then you do the same for the rest of the data that you have. You plot your coordinates in terms of when the time was 8 hours or 9 hours, the temperature was what. So you must be able to draw that and put a dot where they intersect. So that is how you are going to do your graph. Now, in the example, the point where 5 hours and 24 hours degrees intersect on the graph is indicated on the second dot of the graph. Then plot all the points using the information on the table. So that table that I showed you, these are the points that you have plotted where they intersect. Then the next step is to join the dotted lines using a ruler until all the dots have been joined in sequence. So you will use your ruler to join this dot to that one. Don't be in a hurry to make something neat. Then you join that one to that one. You join all the points that you have drawn. Please excuse my squeaky hand because I'm not using a ruler. So you can use a ruler to have a nice, beautiful graph drawn. So you join your lines in this way. So here is a graph that has a line that is joined. Then step number 6, you must give the graph a heading or a caption. So this graph must tell us what are we looking at. And this heading should include both variables. The graph showing the influence or the effect of light on the growth of tomatoes, or time in hours, temperature, what? It must have 2 variables in the naming of the heading. Then in case of air temperature and the time in period of 24 hours must be, okay, in this case, because our table has temperature and it has also time, so it must be mentioned in the heading. So your heading will be something like this. This is a line graph showing the air temperature measured over a period of 24 hours. Then you know that when it was 4 hours, the air temperature was this much. You will be able to get that information from the graph. Let's say 20, 22 maybe. You see, you are able to interpret the graph because you have the 2 variables in your heading. So make sure that your heading has 2 variables. Then, if you are drawing 2 line graphs on the same set of axes, obviously you are going to have a key. Very important. A key must tell us that the dotted line graph shows us it is a carrot. And then, okay, maybe this one, the carrot comes here, and then the dotted line shows us it's the beetroot. Then you will come and be able, or you can even write it on the graph to say this one is beetroot, and this one shows us the carrot. So you see, you have drawn the two graphs on the same set of axes, but you used a key to identify what graph is each, or what graph is this, what graph is this one. So you must be able to use that key. Now, let us draw a line graph. A person took part in an experiment on the eye's response to light. A lamp was placed at 7 different positions from the person's face. The diameter of the person's pupil was measured at each position. So you must understand that statement, what is happening. They took the light, they want to check the eye's response to light, then they put the lamp in at different distances, and then they measure the pupil size of this person. Then the diameter of the pupil's face was measured at each position, right? The table below shows the diameter of the person's pupil when the light was placed at various distances. So, reading the table, when the lamp was placed at 4 meters away from the person, the diameter of the pupil was 3 millimeters. Take note of the units there. When the light was placed at 7 meters away, the pupil diameter of this person was 4.8. So you must be able to read the table first. Then draw a line graph to represent the information given in the table above. So let us look at the first point. When the lamp was at 1, the diameter of the pupil was 1.2. 2, it was 1.8. So let us look at those two. Remember, you are going to identify your variables first. So when you identify your variables, you must tell us this will be your independent variable because it comes on the first column. Also, it is what you can manipulate. It is what the investigator can manipulate. So this here is our independent. This is our dependent because it is depending. So the more you put, the closer you put the lamp, or the further away you put the lamp, it will influence the size or the diameter of the pupil. So this is why it becomes our dependent. Then you draw your set of axes there. This is your x-axis. This is your y-axis. So where do you put what? Then you must decide. The distance from the lamp is your independent. We did say from the previous table. Then obviously, because it is independent, it will come where? It will come on the x-axis or on the horizontal plane. And then the diameter of the pupil will come on the y-axis or on the vertical axis because it is the dependent. And please take note of the units. When you are given units, please write them when you are writing the headings of the axes. Then you come up with a scale for your distances. So the scale is when it was put at 1 meter, 2 meters, 3 meters. So here it's easy. The scale will just be 1, 2, 3, 4, 5, 6, 7. You've decided on the scale for your x-axis. And then you check your data for the y-axis, your smallest and your highest. And then you decide what scale are you going to use. So in this case, we can use 0.8, 1.6, 2.4, 3.2, and 4. So you see, you are moving with 0.4. So you can move in those intervals. The spaces must be the same. The spaces must be equal where you are moving on your y-axis. And then you are going to plot now. When the lamp was at 1 meters away, the diameter of the pupil was here. It was 1.2. It is 1.2 here. 1.2. You see? So you have your 1.2 where the lines are intersecting. Then you plot all your points according to the table, they will come, they will come, they will come. Then you join them together with a ruler. So that is your line graph. But you must also put a heading. So this is a heading, the caption. A line graph indicating the diameter of the pupil of a person if a lamp is placed at different distances from the person. So you see, it has the distance. It also has the diameter of the pupil. Distance, diameter of the pupil. In your heading, you must have both variables. In your heading, it must have both variables. So you see, then you are done. You will get a mark for writing the TLCSP. This is how your graph is going to be marked. For the correct type of graph, if you were asked to draw a line graph, you get a tick for that. And then for labeling the x-axis and the y-axis together with the units, you are going to get 1 mark. Caption with both variables, you're going to get 1 mark. Then the scale for the x-axis and the y-axis, if it is correct, you get a mark. Excuse me. Then plotting. For plotting 2 points correctly, you will get 1. For plotting all points 1, 2, okay, 3, 4, 5, 6, 7, 3 to 7, you will get 2 marks. So for plotting will be 2 marks. So this is how you get your 6 marks for the graph. Then moving on, bar graph. You still do the same. You do the same. Now, the different thing is here. Okay. Right. All of these are the same. This part here, the bars should be of equal width, and the spaces between the bars should also be of equal width. When you are drawing your graph, you don't want your bar graph to look like this. You have this thick of bar, and then this bar is this thick, and then you have this smaller yana bar. And the spaces in between the bars must be equal. The bars themselves, the width of the bars must be equal. So once we see this, you are going to lose a mark for this type of graph. Make sure that your bars are equal and the spaces between your bars are also equal. Then, how to draw a bar graph? Here is, you have the point in numbers. You have point number 1, point number 2, point number 3, point number 4, point number 5. The number of organisms at these points, 10, 12, 8, 8, 4. Now, draw bars to represent the number of organisms found at each of the points along the river. So these are the points on the river. So you will tell us, at point number 1, there were 10 organisms. At point number 2, there were 12 organisms. You see? Now, the bars should not touch one another, as the points along the river have no direct relationship with each other. So this is how you are going to draw your bar graph. Point number 1, there were 10 organisms. So on your x-axis is the point which was, remember, on the first column. It was on the first column, which we see. This was on the second column, which tells us it's a dependent variable. Then you put your points according to your scale, and then you also decide on the scale for the y-axis. You check the highest. The highest was 12. The lowest was 8. So you can use the intervals of 2, 4, 6, 8, 10, 12 up to 14. Then at number 1, you tell us how many organisms. There were 10. Point number 2, there were 12. Point number 3, there were 8. Point number 4, there were 8. So look at the spaces between these bars. It is equal throughout. So your space between the bars should be equal. And the width of the bars itself, the width of the bars should be equal. There is no one that is thicker and there's no one that is thin. So, and then you come up with a caption again to say this is a bar graph organizing the number of organisms found at each of the points along a river. Where did you get this? You got it from the statement. Sometimes you can get it even from the question. They will ask you to say, draw a bar graph showing blah, 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 blah. Then you know that is where you get your heading. Right. Then, how to draw a histogram. Now, a histogram is drawn in exactly the same way as the bar graph. However, the only difference is that the histogram is used when the independent variable is in groups of information along a continuous scale. So the bars will be joined together. So if you are talking about the season and you say season has January, February, March, season 1, then you know for first season, all the data must be joined together, or the bars must be touching one another, because they are information that is on a continuous scale. So how do you know it? The bars are joined together. They are all joined together. So this will be your histogram. This is how you identify. Note that a histogram, the bars are drawn without any spaces between them. So you see, there are no spaces between the bars that I have just drawn there. Now, an example of a histogram. This is a table representing number of pupils in each percentage range. So you say, in the range of 0 to 19, you see it is continuous. This is continuous. It is not just saying 0. It is continuous range to say 0 to 19. We have no people, we have 0 people. But in the range of 20 to 39, we have 5 pupils. In the range of 40 to 59, we have 11. So how are you going to put this information in a histogram? So this is how you are going to do it in a histogram. Now look at the range in percentages. So here you will have your 0 to 19. Then here at 5 you will have your 20 to 39. This brown one will indicate your 40 to 59. The amber one, it will be your 60 to 79. And the blue one, the blue one is 80 to 100. So they did not write them, but you know, you see, and you see all of these bars, they are clustered together. There's no space. So here there's no bar because it is 0. We don't have learners here, hence it starts growing from here. So, and then your bars are stacked together close to one another, no spaces in between. And then you can come up also with a heading to say this is a line graph representing the number of people in the percentage range. So you see, you take the heading of the table as it is. You can put it as the heading of your graph. You see, just like that. Then you get your full marks. Now, this is where most of you are also losing marks. How to draw a pie chart. A chart shows data as a percentage or as a relative proportion of the total of a circle, meaning in the 360 degrees, what will be the proportion of a particular data. Then you say you must divide that into sectors. You draw a pie chart by dividing a circle into sectors or the slices. You can think of a pizza to represent each item. So you must make sure that whatever data you have, you convert it to be able to fit into a circle, because a circle, you know, is 360 degrees. Degrees, not Celsius, just degrees. You must then convert everything to 360 into angles. Then you will be able to plot them. Let us take a look at those steps. What you need to do when you're drawing a pie chart. To draw a pie chart, we need to represent each part of the data as a proportion of 360. You see? Why? Because a circle is 360 degrees. So everything must be converted to 360. So if you have a data here, this is the table, the table below shows the percentage of carbon dioxide emitted by different sectors in a certain city in South Africa. So already you have the title of your pie chart from the title of the table. Then you take each sector. The transport sector is emitting 25%. The residential sector is emitting 27%. These are the emissions of what? Carbon dioxide. Industrial sector is emitting 15. Commercial is emitting 28. And then other sectors combined together, they are emitting 5%. So you have all this information. Step number 1, what are you going to do? You are going to add all the data in the table together. In this case, you're going to add the 25 plus 27 plus 15 plus 28 plus 5. So here, 25, 27, 15, 28, 5. All of them are equals to 100. Right. Then what do you do with this information? You see, all of them together, it is 100%. Because this was given in percentage, yes, you would expect the total to be 100. But you can't just go to a pie chart and include, say, 25, say, 30, say, 70, 25, say, 27. You need to be sure from this 100, how are you going to convert this 100 into the degrees? So the next step of what you are going to do, right? So you are going to do the following. Step number 2, do the following calculations to calculate the angles of each sector. Now each value or the total multiplied by 360 is equals to the number of the degrees that you want to put in your pie. So you are going to put in your pie chart. So what is happening here? You take the value, you divide by the total, you multiply by 360. Every time when you are converting to something, you must multiply by it. So right now we want to convert to 360, then we multiply by 360. Now let us look at our example. Transport, we said it is 25. So our 25 is our value divided by the total. So value divided by total multiply by 360. Then when you do that on your calculator, it will give you 90 degrees. So once it gives you 90 degrees, you must be happy because on your pie, you already know, you draw your Cartesian plane like this, you know that this angle here is 90 degrees. So already you see that you have your transport sector here that is emitting 90 degrees. And then you have the total of residential, which is 27, divide by the 100, which was the total of all of them. You multiply by 360, you get 97.2. Please make sure that you get the value that you want to convert, divide by the total that you got of all of them, then you multiply by 360. It will give you that those numbers or those values converted now into angles. So you see even the number, the answers are written in degrees Celsius because you were multiplying by 360 there. The industrial sector will give us 54 degrees. Then the commercial sector will give us 100.8. Then other sectors will give us 18. To verify your answers, you can also take all of these angles and try to add them. They must give you plus minus because when you are doing calculations where there are commas, you might find yourself that you rounded off the number. So it must give you plus or minus 360. Then you know that it is correct because the proportions will all fit into a pie. So let us check in our example. Then you must have this. You must have a compass to draw the circle. So this is your compass. And then you must have a protractor. So this is your protractor. So protractor. Then this is your compass. So you can even use the protractor itself to draw a circle, but you must be careful when you draw, you don't put these values here of the zeros. So your angle must start from 0 and 180, not this part that does not have the values, so that it can be a complete perfect circle. Otherwise, you can use a compass to draw your circle. So here, now we are putting them using our protractor to proportion the section, because the protractor has the angles in the degrees. Then you just put the lines on the degrees and you put them. So your first sector was your 25%. Remember, this was done with a computer. Whereas if it was you, or if it were you, it would have been simpler to start with your transport in this way. Because you know that a circle will start like this, start at 0. Here is your 90. Here is your 180. These are degrees Celsius. Here is your 270. And at the end is 360. So for 90 degrees, it's easy, because then you can just do this and say, this is your transport. And then from this one, you know that it's already 90, but because you need the 7, you take your protractor and then you add up to 7 here, and then you draw a new line, then you erase this old line, then the whole sector will represent the residential sector. Then you start proportioning your other sectors that are remaining until your complete angles of 360 are fully represented. So you will see, you can write them inside. Now use the values that you've calculated. Remember, you must also have a heading: carbon dioxide emissions in different sectors of a city in South Africa. So you can even add that, because it was given to you in the table. Right. Then if you are not labeling the keys, you should make a key which explains what each sector represents. So some of you might have a key on the side to say the dots will be our transport. Then you have the stars inside, you will be representing residential. Then you have maybe the circles. The circles will be representing industrial. So you see, then you do it like that. Then transport, you put your shades like this using a key. And then your residential, you put your stars according to your key. Then we know that this is residential, even if you did not write it inside. But using the key, we are able to identify what is in each sector. Then your industrial, just for complete graph, then you will have something like this. Then we know. Or you can even use colors. So you can be creative with this. Please make sure that you collect all the 6 marks also on your pie chart. So this will be your pie chart of colors. So if you use the key using the colors, then we know this green color represents transport. You see, this is transport. You did not write it, but we can get it from the key. The blue one is other, which is 5%. Residential is this one. Then this one is industrial. Then commercial is this one. So you can use either way, whichever way that works for you, depending on the resources that you have when you are writing. But please remember those instruments that you need. Then, what is the criteria for marking a pie chart? You are going to mark the correct type of graph, the title of graph, carbon emission of each sector in South Africa. So carbon emission and sector, they are the variables that are needed in this case. Then you get 1 mark. For the calculations, if you've shown all your calculations, 1 to 4 calculations correct, you get 1 mark. All 5 calculations correct, you get 2 marks. Then the correct proportion of each labeled sector. So when you see the sectors that you've converted, it must be correct. So 1 to 3 sectors are correct. Do you see that even if you had drawn only that 90 degrees, you were still going to get 1 mark? Because it says 1 to 3 sectors correct. 1 to 3 sectors, you get 1 mark. 4 to 5 sectors, you get all 2 marks. Please make sure that you don't lose the marks unnecessarily. Even if you don't have the apparatus, you can draw a circle and get a 90. You can also get the type of graph. You can also get for the title of graph. The title of graph, the type of graph, and the proportions, even if they were not correct but just one, you can get 1 mark. So you can get 3 out of 6. Do not leave it, even if you find that you do not have the instruments that are required to draw a pie chart. Then we come to diagrams. Life sciences has diagrams. All the diagrams have rules. You don't just draw a diagram. So let us check what needs to be there in a life sciences diagram. First, each and every diagram must have a title or a caption. I used to say to my learners, if you have drawn something and it does not have a caption, I will just write potato on top of it. Because you can't stop me from saying that thing is a potato when you did not tell me what it is. So you must give a caption always. A diagram of the eye, diagram of whatever. Write the caption or the title. Then you use a pencil to draw a clear, smooth lines. When you draw the lines for labeling, you must use a pencil. Remember, you are using a pencil to draw the diagram. Then you use a pencil to draw the clear, smooth lines. Then, when you label, you must use ink, a pen. You must write your labels in pen all the time. Then, some of you draw small diagrams. You are asked to draw a diagram of an ovum. You are going to draw something like this, a small ovum with the jelly layer, the cytoplasm, and then the nucleus. So how many marks are you expecting to get from that? So it must be large enough so that all the structures are visible. And I've just chosen a simple diagram. Sometimes you can draw a diagram that we need to see. Let's say the sperm cell. So we need to see those labels, those diagrams also visibly enough. So it must be large enough. Don't spare the space. Then the diagram must be positioned in the center of a page. So you don't draw your diagram here or what. It must be at the center of a page. And then it must be two-dimensional. It must show the length and the width, and you do not shade. No shading at all. It must be clear. And let's go back to the pencil lines. When you draw your label lines, your lines must not overlap, and they must be of the same length. They must stop at the same length. So your lines cannot go crisscrossing like a spider and doing what? They must all be on one side, and they must be of equal size and no overlapping. You would rather go to a structure. Let's say this is the structure that you want to label. You see the structure that I'm making in the middle there. You would rather go to that structure and get a line to bring it to the rest of the lines that are on the right hand side. Then your diagram is going to be neat. Please, 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 no spider crossing, no crossing of the labels. When you draw your diagram must have all the lines on one side, drawn with a pencil, and the labels with a pen, and no or crossing. So this is an example of a diagram that you can find in life sciences. So look, draw the lines with a ruler. Your lines must be clear. Now the lines must not cross each other. This is what I was talking about. The lines must not have arrows. So you don't find yourself drawing lines with arrows like this. No, 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 no. No arrows. Arrows are incorrect. Just plain straight lines. Then the lines must touch the structure that is labeled. So it must show us that here, this is what I want you, the alveolus, this is what I want you to label. The bronchiole, you must see it. This is what I want you to label. And here, oxygen diffusing into, I want to see what does it want. Carbon dioxide diffusing out. You see, so we want to see the red blood cells. It must point exactly to the structure that you want to label. Then the lines on one side of the diagram, they must be few. So you must be lines on one side of the diagram. Then label lines must be aligned neatly and preferably one label below the other. So they must be aligned and one below the other. Do you see how neat this diagram is? It is, you are able to read the diagram. So when you draw your diagram, you must keep these principles in mind. So sometimes you are asked to draw a table again. Draw a table differentiating between the two. So what are the rules for drawing a table? First, you must have captions in each column. Can have a caption also for the table itself to say, remember the tables that we've just worked with right now, they had a caption to say, this is the table showing the carbon dioxide emissions of each sector in South Africa. Then you see it is a table with a full description of what type of data are you going to get from that table. Then you can have also the column captions to say, in these columns we have the humans, in this column we have, so this is column number 1, you have your humans, column, and then African apes. Then you start writing, there are no rows. Some of you don't even have this line, there's no column. Then you end up losing the marks because a table, for it to be complete, it must have rows with columns. So what gives us the rows is this, the lines that you have. This is our column. Column 1, column 2. Then divided into rows. Row 1, row 2, row 3, you see, row 4, and row 5 and 6. Then you have your full table. Then this thing, we can call it a table, right? Then the same factor must be compared in a row. So you compare cranium size, large, cranium size, small. You don't say cranium size here and then this size by the starting value. So you take, if they say from 2000 and 2006, you take the starting value, you divide the difference, you find the difference first, then you divide it by the starting value. This is your first value. So by doing that, let's say the following table is a summary of the rhino poaching incidents for South Africa from 2000 to 2010. With what percentage did the poaching of rhinos incidents increase in Northwest since 2008 to 2010? So 2008 is this one to 2010. So you take 44, you minus the 7, you get the difference. Then that difference, you are going to divide it by the starting value, the first one. Right. So how do you do that? Here, 44 minus 7 multiplied by 100 is 7. Then you will say it has increased by 529%. So that is how you calculate your percentage. Then how to pass grade 12? These are just the tips that you need to be able to do to pass your grade 12. Right. So please make sure that you go, you study, you revise using all the materials that you did, and be at school with your friends and study. So I wish you nothing but best of luck for your examinations, and thank you so much. So I would like to say thank you so much for watching today's lesson. I will see you again next time. Goodbye.